Welcome you all to this Cryo-EM course. Last class we discussed about how we can prepare the samples, right? We already discussed about protein purification, and then there are three different types of protein purification we can use it, starting from affinity purification, ion exchange purification, and then size exclusion chromatography. And also we can purify our DNA, RNA, or we can synthesize this RNA, DNA, and then we can form the complex also. Also same time we can prepare the lipid, all liposome different concentration, and then from this liposome we can incubate with our protein. If protein has some affinity towards lipid, then we can form a lipid protein complex and we can image using the negative staining. Also we discuss about the different types of grid, that finder grid or normal grid. Normal grid means how many squares are there. Based on that we are telling that it is a 200 mesh grid or 300 mesh grid or 400 mesh grid. So now today we are going to discuss about the negative staining, how we can do the negative staining. We will also discuss about the metal shadowing for especially for DNA. Nowadays these techniques we are not using very frequently, but that is a good technique how we can use the metal shadow in technique to coat the DNA, as well as we will discuss about negative staining TEM data, how we can use this negative staining TEM data. So what is the application of negative staining TEM data and how useful it is. And then last one I will show you some bad images and some good images. So we can figure it out that when we are doing negative staining, is this data good enough for publication or for my research thesis, or do I need a good quality data, and how we can get good quality data. So I will show you some TEM images and describe which one is a good data and which one is a bad data. So let's start with our slide. So based on the today's topic, we are going to focus on TEM sample preparation and carbon coated grid preparation and negative staining procedure. So first we want to discuss about the TEM grid. Last class I already discussed about the TEM grid, that you can get a different types of TEM grid. So where you have a different type of mesh system. Now if you just look at this TEM image, this is a TEM grid. This is a enlarged view of the TEM grid, where you can see this is the metal bar, where this part it will help you to hold this grid, because this grid overall diameter will be 3.05 mm, and the grids are very fragile. You have to handle it very gently, and these are the square area where we can do our imaging. But normal grid, whatever I showed you last class, it may be commercially available, that grids are already carbon coated or Formovar coated. So or that will be an empty grid. Most of the time previously we are just purchasing the empty grids, and we are coating our grid with some kind of plastic, and then on top of that we are putting the carbon, and we are using those carbon coated grid. So you can easily make those carbon coated grids in your laboratory. Also those grids are commercially available, so you can purchase it also. But in this class I will give you an idea how you can prepare those kinds of carbon coated grid in your lab. You don't have to purchase those kinds of grids all the time. Now here you can see this is a grid where you can see multiple squares. Based on this number of squares we can tell that it is a 200 mesh or 300 mesh grid. Now if you come here, you can see in this grid there is a two different layers are there. So one layer, it is a Formovar. Formovar, what is the Formovar? Formovar is a thermoplastic resin, that is basically are polyvinyl formals, which are the polymers formed with polyvinyl alcohol and the formaldehyde. as a copolymer, is a polyvinyl, copolymerize with the polyvinyl acetate. So, that is a chemical and it is forming a very thin transparent plastic layer. So, this is basically Formovar. There are other compounds also you can use it, that is that form a plastic coating, that Formovar or chlorine. So, those compound it will coat the grid and then on top of that we will put a carbon coating. So, mostly in carbon coated grid there will be a plastic coating and then amorphous carbon will spread all over this grid, and this is a holy carbon grid. We will discuss about the holy carbon grid in later. So, basically this holy carbon grid we are using for cryo sample preparation. So, if you remember properly, there are two different types. One is room temperature negative staining temp sample and another one is quantifoil grid. It is for cryo sample preparation. For room temperature temp we are mostly using that carbon coated, continuous carbon coated grids. So, here we are showing a holy carbon grid. There are also different types of holy carbon grids are possible, or lacy carbon grid is also possible, which we are using for cryo. So, in next class when we will discuss about the cryo sample preparation, we will talk about this one. Now, here you can see there are some kind of boxes where this is the lid, where you have the small hole in all these boxes. So, this grid can go inside this individual square, those are called the grid boxes. So, different types of grid boxes are possible. Sometime you will get a 25 grids or sometime you will get a 100 grid box, sometime this circular grid box, and all the time when you will do your negative staining and you have to store your grid in these grid boxes. Most of the time negative staining grids are good for good to go for long time. So, that means if you prepare a grid, it will be good if you can preserve that grid properly. It will be good next 1 year, sometime 2 years or 3 years also. If there is a not too much humidity or moisture is deposited, this grid will be really good. So, you have to preserve those grid properly. So, that is the reason this grid box you have to label it properly, otherwise if you are continuously doing negative staining imaging and storing this grid, and if you do not have the proper labeling, it will be very difficult to find out what grid you prepared almost 6 months ago or 1 year ago. So, that is the reason these are the grid box. You have to select what kind of grid box you want and you have to document those things properly. Sometime for negative staining, one day we are checking some 15 grids or 20 grids, or maybe you just consider in 1 week we have some 30-40 grids we checked at different sample condition. And then if we store those grids properly, then we can go back and easily re-image using those grids. If we do not label it properly, it will be a difficult task. So, that is the reason this is a grid box and the labeling is very important. Now, you can see this is a cryo grid box where you can store only 4 grids, and every time there is a plastic cover, that lid which is covering that grids. So, there will be this entire grid box will stay in liquid nitrogen, and there will be so this grid will be covered by this plastic lid that will help us to avoid the ice contamination. And again, using this screw, you will tighten this grid box in such a way that grid cannot come out from this place. So, we can store this grid box in liquid nitrogen for long time. After freezing we can check these grids after 6 months. There is no change will happen if we can preserve these things properly in liquid nitrogen. Now, I will go to the next slide and then I will show you that this basically is taken from a book chapter that I already discussed last class, that is the specimen preparation for high resolution cryo-EM. So, basically those things are applicable for cryo-EM, but same time similar approach we can take it for negative staining also, and that is the reason this book chapter is good for when we are starting the sample preparation, how we can prepare the sample and which particular area we should focus before we start handling those grids. So, here I am showing you a gold grid, and the same gold it is like a copper grid, and then there is a different type of just the color will be different because it is a gold coating. If we are just using normal carbon coated grid for negative staining, the this color will appear like a brown. If you just look at this one, this is the color of the copper, and this is a gold grid. And you can see now, if you focus on the, if you just enlarge one particular area of this grid, you will see this kind of square, where this is the major metal bar and then this is the carbon area. So these grids, as I mentioned earlier, it is mainly focused, this book chapter mainly focused on the cryo-EM. That is why we are using a quantifiable grid where you can see a uniform sizes hole. This part we will discuss in the later stage. Now if you look at this square, you can see this kind of copper or gold bar, and the middle your sample will stay here. This is the area where you have the formovar and the carbon coating, and electron beam can go through this sample and we can image our sample. Now the next part is an important part here. Why it is so important? In this book chapter, authors specifically mentioned how you should handle your grids. And so to handle this kind of grid, because these grids are very fragile, if you have to handle these grids properly. If you cannot handle it properly, it will damage the grid and it will definitely damage your sample preparation technique. So here, if you just go back to the previous one, you can see there is the metal bar, this particular area, it is quite thick. So you can use your tweezers to hold this grid in this particular area, not anywhere at the center or middle of this grid. Why we cannot do that? If you come here, you can see whenever you are holding your grid at the middle or center somewhere, it is damaging the surroundings area. It is damaging the surroundings carbon and formovar area, and your grid will be not flat anymore. So here we are showing a very sharp tweezer we should use. We should not use any blunt end tweezer, and that is going to damage our grid. And also when we will try to hold our grid, we should not hold our grid like that, where this tip of this tweezer is going to touch that individual square of that grid. So generally outer ring we should try to hold our grid gently and do whatever it is required. So that is the first step we should follow. So now if I go to the next one, how should we pick up our grid? So here one important part it is showing, in a petri dish there are multiple grids are here, and as I mentioned that grids are the thickness is very little. So you have to handle it gently, and diameter will be 3.05 millimeter. Now when you will pick up your grid using a tweezer, you should not directly hold that grid from the side. So you gently use your tweezer in such a way that you can pick up your grid from the side, not from the center. So if you just try to pick up your grid from the center, what will happen? It will bend the grid. So if you look at my hand, if this is our grid, if you consider this is a grid in the petri dish, and if you are trying to, this is your tweezer. So if you are trying to take the tweezer and trying to pick up this grid, what will happen? You will bend that entire grid. So entire grid will bend. Or if you try to take this part, what will happen? This particular part will be bent. So you cannot try to pick up your grids like that. What you have to do? You have to go little bit slanting condition. This is your tweezer, and then you try to hold this grid, and whenever you will try to hold, you don't try to hold your grid from the center or the middle, or so it will be near the periphery of this grid. You will try to slanting your tweezer, go there and hold your grid, and then transfer to a different place. So this is the way here. So basically we are showing that how you should pick up your grid. So you should go as a slanting condition and pick up that grid. So you should not pick up your grid whatever way you want. And now the next step we are going, and wow, okay, so one more important part is that, so the use of gloves and plastic storage dishes result in the accumulation of the static charges. In the image, a charge grid is standing on end. So here one more important part we are showing that, okay, so if you just use the gloves and the plastic petri dishes, it can generate a static current. So, and the static charge, you can see some of these grids are standing like that. So, the panel B is showing that recommended handling procedures including glass containers, no gloves on the hand holding the tweezers and a wrist grounding strip to prevent the accumulation of the charge. So, here you can see the hand is touching. So, here also you have a gloves and hand is not touching the floor. So, that is the reason there is a static current it is happening and grid can float here and there. So, here you can see the hand is stable on the platform and there is no gloves, and you are slanting way you are trying to pick up those grids. Okay, this is one of the very important part when you will take those grids. So, when you will take those grids, the next step it will be, so we have to do the glow discharge. So, glow discharge, it will help us to make our grid hydrophilic. So, let us discuss about that, how we can make that grid hydrophilic and why it is important. If we do not make our grid hydrophilic, what kind of problem do we face? Before going to that, we should know little bit about that when we started that negative staining TEM imaging. So, negative staining TEM imaging we started long back. So, when we, if you remember in 1931, first Ernest Ruska discovered that electron microscope, then scanning electron microscope, and gradually it evolved. But similar time scientists and researchers are using negative staining technique, because that time there is no cryo-EM techniques that you can use it at cryogenic condition. One condition you remember, that whenever we are talking about negative staining transmission electron microscope, that means we are using some kind of heavy metal staining solution to stain our sample. In that case, negative staining means we are staining the background, not the protein sample. But in cryo condition, we are not using any kind of staining solution. So, that is one of the major differences, that our protein sample will stay in a native condition all the time. Now, if we come here, you can tell that the basic negative staining techniques has stood the test of time, and since it introduced as a laboratory procedure in 1959 by Berner and Horney, as which is fully acknowledged by Harris and Mohn. So, in these techniques what we are doing, we have a carbon coated grid. Carbon coated grid means there is a formova coating, then there is a carbon, and then we are adding our sample. We are using this sample to stain with a heavy metal staining solution. There are different types of staining solution is possible to stain our sample. Now, what kind of sample we can visualize by negative staining TEM? Here you can, if you just come here, you can see any kind of biological sample we can image, but it should be thinly spreaded on top of the grid. If you remember last class, I was talking about that there is a procedure that when we can use normal room temperature TEM or cryo-EM, when the thickness of the sample will be below 250 nanometer. If it is less than 100 nanometer, that will be the good thing. But if it is higher thickness, that is above 200 nanometer or 250 nanometer, probably it will be, we can use it for mainly for cell or tomography purposes. So, this thinly spreaded biological sample is an important part, and then wide range of samples like virus to any single molecules, like that any macromolecular assemblies. So, subcellular membrane fraction, lipid suspension, toxin lipid membrane interaction, everything. So, we can use negative staining to get any kind of sample. or visualize any kind of sample using that negative staining technique. So, let us come back here and then let us take a look that how we should use this negative staining technique. Before that, we want to discuss little bit about that how we can do the carbon coating in our laboratory, and that will help us to go for the next step, and we can make those carbon coated grids in our laboratory. So, in this particular slide, what we are trying to show you the preparation of the Formovar film coated grid. So, what is that Formovar film coated grid? So, here there is a container where we have a Formovar solution. You can see this is our Formovar solution. This Formovar solution, there is a clean slide. The slide should be as clean as possible, wearing a gloves, and then gradually dip that slide inside this solution. When you will inside that solution, this Formovar solution will attached, it is attached with this clean slide in both the sides. Why cleaning is very important? Because this Formovar will form a very thin layer of plastic, and we have to isolate this thin layer of plastic from this glass slide. So, if there is any contamination or any sticky material attached with that glass slide, it will be very difficult to separate this plastic coating on top of this glass. That is the reason initially it is important to clean the glass slide properly and then dip this glass slide inside this solution. After holding for some time, when this glass slide is completely soaked with this solution, you can take out this glass slide and keep it on top of a filter paper for quite some time. So, when you will keep this glass slide on top of this filter paper, excess solution will be blotted away by this filter paper, and a thin layer of solution will be deposited, and slowly it will be completely dried and fixed on this glass slide. When it is completely dried and fixed on top of this glass slide, we can wait some 30 minutes, sometime 45 minutes, sometime 1 hour. When this glass slide is completely dried, we can take this glass slide, keep it on top of a filter paper again, and then we can use a very sharp blade and try to cut that side of this Formovar plastic. So, here the Formovar plastic is coated all sides, and now if we want to isolate this plastic coating on top of this glass slide, we have to separate this plastic coating from the other side of this Formovar. That is the reason we are using a blade to cut those areas gently. So, this will form a rectangular shape. When we have this particular cut, then take this one gently and immerse inside a water bowl. So, here there is a container where we have the water, and then you keep the glass slide 45 degree according to this container. So, here this container and this gradually immerse this glass slide at a 45 degree angle. When you will immerse these things as a 45 degree angle, this particular part is already detached from the glass slide, and when you will gradually dip this one, this plastic will start floating on top of this water. And if you slowly go, you should not be in a rush. This should be holding this glass slide using a tweezer tip, and do it gradually, very slowly. You should immerse this glass slide, and this plastic will start floating, and then gradually you go all the way down where this particular part will be detached from the glass slide, and glass slide will go inside the water. When you will gradually drop that glass slide, you do it very gently, otherwise it can damage this plastic layer what is floating on top of that water. when this is floating on top of that water, then you can slowly place your grids on top of this plastic layer. If you put the grids on top of this plastic layer, what will happen? You have an empty grid, and that empty grid you are trying to coat with that plastic. So, you are going to place the grids on top of this plastic layer. Now, if I go to the next slide, you can see it here. What I am trying to say, here you have a plastic layer. So, this is the container where we have the water. In the last slide, I already showed how this plastic layer will float on top of that water. Now, very gently you place all your grids on top of that plastic, and you have to do it very gently. So, using a tweezer, you have to hold that grid gently and place it. If your hands are not stable, you are going to break that plastic layer, and then it will damage the entire preparation. When you place your all the grids on top of that plastic, there is a paper, specific paper. You can take this specific paper and place at top of these grids, and this paper will be soak the water, and then it will spread like this. When it will spread, gently cut the rest of the plastic layer, and then pick these things from the water and keep it for drying condition for at least 1 day. So, this is the procedure. You start from here, you use this kind of glass slide, and then when this plastic formova coating will float, you will place all the grids on top of that, and then you put a paper. That paper, when you will put this paper, what will happen? This in between this paper and the plastic layer, you have all the grids. The paper will be flat after half an hour, 45 minutes, and then cut the rest of the formova plastic from here using a blade, and then pick up these things very gently. When you are putting this paper, paper should stick with that plastic, this formova properly. If it is not stick with the formova properly, then when you will pick up this paper here, what will happen? The formova and grid and the paper will be detached, and your entire procedure will be damaged. So, you need little bit practice to do this exercise, but this is how you can coat your grid with some formova coating or chlorine. When you have this grid coating is ready, you can take it for the next step for doing the carbon coating, because carbon coating is also important, because only this formova coating, it is a very thin plastic layer, as I mentioned previously, and when electron beam will pass through this one, it will damage the plastic immediately. So, that is not a very good situation. That is the reason if you do a carbon coating, it will give you some kind of stability, and that is one of the reason. First we do the formova coating, and then we do the plastic carbon coating, and then we can use those grids for negative staining purposes, where we can use that different type of staining solution. So, if you are interested, you can follow this reference book. You can get how to prepare that grid, formova coated grid in the laboratory. In the next session, next class, I will talk about how you can coat those grids with a carbon layer, and then we will also discuss about why we have to do the glow discharge before we add our sample, and then we will talk about negative staining procedure. Thank you all for your attention.